Hello, everyone, and happy Friday. My name is Cindy Beslever, and I am a member of the Scleroderma Foundation Board and the Scleroderma Foundation Rocky Mountain Chapter Board. Thank you for joining us today. This year is the Rocky Mountain Chapter's 20th anniversary. And to celebrate, we have launched the Imagine campaign, which we hope to raise $100,000 to launch the Scleroderma Foundation Rocky Mountain Chapter into the future. 1/3 of the funds raised from this campaign we have earmarked for peer-reviewed research, scleroderma and fibrotic research through the Scleroderma Foundation's research grant program. The Rocky Mountain Chapter Board of Directors has pledged to match all donations up to $8,000 raised by through the Imagine campaign in the month of March. Well, goodness, I'm stumbling over that because I am so excited about that. Thank you, board members. Joining us today is Dr. Carol Feghali-Bostwick. I would like to introduce her to you. She is a professor of medicine and serves as the SmartState and Kitty Trask Holt Endowed Chair for Scleroderma Research at the Medical University of South Carolina. She also serves as vice chair of the Scleroderma Foundation Board of Directors. And before we jump into talking to Dr. Feghali-Bostwick, she is in her lab this afternoon. So she is going to give us a little tour of the lab and where it all happens for Scleroderma research. So I'll turn it over to you, Dr. Carol Feghali-Bostwick.
Thank you very much, Cindy. I'm thrilled to be with you guys today. I'm so impressed and so wowed by what the Rocky Mountain Chapter is doing to support research. And I'm very grateful, as are many people who want to see research on scleroderma advance. So let me switch this around to show you. We're heading into the lab. So this is what a typical research lab is. As you can see, with a lot of reagents. And we're going to take a look at some of the type of assays set up. Like this is a setup to look at proteins, different centrifuges. This is Joe. He's a graduate student getting his MD and PhD here. Joe is imaging what we call Western blots. He's looking at proteins on this piece of equipment to measure how much protein is in cell samples. So in addition to typical work on the bench and imaging and looking at RNA and protein and a variety of reagents like that, we also do a lot of work with cells. So we grow a lot of cells. We do that for our experiments to test the effect of agents that cause fibrosis and agents that may prevent fibrosis. This is, for example, our microscope setup that we use for imaging to look at pictures of cells. We have a special room with the lab that we use for growing cells. It has to be a separate room where things are kept clean in sterile conditions. And these are the areas where we work with the cells. They're called tissue culture hoods. And then the cells are grown in these, what we call incubators, where tissues and cells, as you can see, are maintained at what we call body temperature. And actually, what I'm going to do is show you very quickly what it looks like to grow cells from tissues.
Dr. Feghali-Bostwick, are those from scleroderma patients? Are those sample tissues from patients?
We have both. This particular one I'm showing you is we're growing fibroblasts from small pieces of skin biopsies that are actually from a healthy donor. So you always want to compare a healthy donor to patients with scleroderma, the response to see if it's the same or not. We can obviously image the cells. This is the nutritious media that we grow the cells in. It's a liquid that has all the essential vitamins, amino acids that cells need to grow and thrive. And what's really, to me, has always been amazing, even though I've done research for a long time, is that we can freeze the cells that we get from donors and patients for our research. And what we can do is freeze them down in temperatures that are just tremendously cold. So we freeze them in these, what we call R2D2 units, but these are, I don't know if you can see the gauge, minus 143 degrees Celsius. That's colder than minus 200 Fahrenheit. And we can keep them in there frozen for years, and then we can take them out, thaw them, grow them where I showed you with the nutritious media, and they'll grow again and divide and proliferate, and we can use them for research again. So we can always use them for an extended period of time. In addition, we obviously have specialized equipment to detect levels of RNA in addition to levels of protein. And the bench area in general is open where people can do their experiments. And then nearby, they have desk areas where they could sit and analyze their data, write the papers for the findings and things along those lines. So this is what a typical lab looks like. There's Elisa. I'm going to show you the view walking backwards. So a lot of different things to consider in the lab, obviously. It's Friday evening here, so not everybody is here at this point, but you can tell there are people who work into the evening to do research on scleroderma.
How many researchers do you currently have working with you in scleroderma research?
So right now, just in my group, there are about 10 people at this time, and they rank different ranks from students to postdoctoral fellows, to junior faculty, to research specialists. So they're all at different levels of their training.
Interesting, very interesting. It's great to see where it all happens.
There.
Well, should we continue?
Yes, absolutely.
Like to move? All right. Okay. Well, can you tell us what is happening in the world of scleroderma research today?
Well, interestingly enough, today a lot is happening. I wouldn't have said that maybe 20 years ago because the pace of research was not quite as fast 20 years ago. But currently, with the available technologies and tools that we have, research is actually advancing at a very fast pace. And so there are a lot of discoveries. Research on scleroderma is being highlighted in very important high impact journals and a lot of publications. Actually, the number of publications on scleroderma has dramatically increased over the past few years.
What do you feel is the most critical to finding a cure for scleroderma and fibrosis or other fibrotic diseases?
I think what's critical is to maintain the research. We don't want to lose the momentum. Like I said, research has really been moving at a faster pace. So we certainly don't want to lose the momentum of the research, but we also want to ensure there's a pipeline of researchers to continue research on scleroderma. That's why the Scleroderma Foundation invests in new investigators, supports established investigator research, but also new investigators to ensure that there's a pipeline of researchers for the future. And actually, I'm very excited to announce that for the very first year, the Scleroderma Foundation has just launched also a fellowship award program for graduate students to try and capture investigators in scleroderma as early as their graduate education.
That's great. If we can get them early and keep them in scleroderma research, that would be great news for us. Exactly. Can you tell us a little bit about how that program works and how the grants are awarded?
So the Scleroderma Foundation, and if you want more details, you go to scleroderma.org, has a very unbiased peer review program for grants. Grants are submitted, usually the deadline is mid-September. The grants are then assigned to reviewers, peer reviewers. So these are experts in the field who themselves did not submit grants so that there's no conflict of interest. They review the grants. They prepare a long description of the grant with what the strengths and weaknesses are of the grant, and they give the grant a score. And then in December, we hold a meeting of all these peer reviewers, and everyone around the table gets to participate in the discussion of a grant and gets to score it. And the top scoring grants, so this is all based on scientific merit, end up receiving funding from the Scleroderma Foundation. And again, those are established investigator grants and new investigator grants. And we've had before SCORE grants. These are multi-center grants. So we designed these grants intentionally to promote collaboration across centers in the US, across scleroderma centers. And it is very likely that we will have those again.
Great. I know you all, even if you're not working together, I know you're all in close contact with each other, all the scleroderma researchers across the country. So you are a past recipient of one of the Scleroderma Foundation grants. Can you tell us how that has helped advance your research?
Right. So I actually received my grant from the Scleroderma Foundation in 1999 when I was starting my independent career as a scientist. Obviously, now being on the board of directors, I don't apply for grants from the Scleroderma Foundation because that would be a conflict of interest. But in 1999, I did get my very first independent grant, and it was a grant to study scleroderma in twins. So to do a twin study, which we did and published on, and it's been the only twin study of scleroderma in the world. So the Scleroderma Foundation really launched my career. It was my very first grant as an independent investigator. And we hear that a lot from our new investigators, that the grant that they get from the Scleroderma Foundation at the beginning of their career is what ends up launching their career. Often it's what keeps them in research because the pressure is on that if you cannot get grants, you are not allowed to do research. And so getting these grants allows them to start their research, generate data that then they can use to apply for larger grants from the National Institutes of Health.
Great. And you mentioned something, Mia, a few weeks ago that I wasn't aware of, that you researchers all have to pretty much get your own funding. So that's why it's important to keep raising money and funding the research.
Absolutely. I think a lot of people don't realize how research works. You actually have to bring in the funding to pay for all the experiments, to pay the salary and fringe benefits of everybody in the lab doing the experiments. And money gets brought in to pay what we call overhead, you know, the electricity, the housekeeping service, everything else. So researchers have to bring that in through grants to be able to do research, at least in the US.
Is there anything else you'd like to add or share with us today?
Yes, I would like to emphasize how critical the Scleroderma Foundation has been in all this. We fund a large number of grants. We have sustained the careers of established investigators. We have launched the careers of new investigators as a foundation, but also how unique the Scleroderma Foundation is, because there is no other organization in the US that supports patients and caregivers, that provides educational programs, promotes awareness, and funds research. So really the foundation does it all. And the Scleroderma Foundation does not do it in isolation. We would not be able to do it without everyone, all the chapters, support groups, the patients, the caregivers. You can find a lot of information on scleroderma.org, but I just want to emphasize to everybody how critical the foundation has been in our lives, whether we're scientists, whether we're patients, whether we're caregivers, or whether we're just your average person out there who needs to know more about scleroderma, as well as a physician who has never seen a scleroderma patient, because the foundation also does physician education programs. So the foundation is trying to take care of all these aspects of its mission and is doing so extremely well, because everybody who's involved cares. The staff at the national office is like a family. They all do this because they genuinely care. Everyone at the chapters at all levels, from the boards to the volunteers, to the employees, the caregivers, the families who give so much in support of the patient with scleroderma in their community, and obviously the medical and scientific community that continues to persevere and do research to try and advance understanding of scleroderma and identify new treatments and new therapies. So it takes a village. It does really take a village, but I feel like the Scleroderma Foundation is the heart of that village and is the anchor for the village.
Well, thank you, Dr. Carol Feghali-Bostwick. We do appreciate you being with us today. And I have to echo those same sentiments. I've been involved with the foundation for more than 20 years, and it definitely is a family and people working very, very hard to find a cure. So we appreciate all that you and your colleagues do. And so you heard it, folks. We sure would appreciate your help with reaching not only our campaign goal, because that's not what is important. The money, what's important is that that money will fund the important research that all the researchers are doing. And again, thanks to our board, chapter board, who is matching the first $8,000 to be raised this month during the month of March. So you look in the chat, you'll see where you can go to donate. We sure would appreciate your support. Stay healthy, stay well, and thank you very much.